Hey everyone, welcome back. We are diving deep into peptides, proteins, and antibodies today.
Oh, those are fun.
Specifically,
Yeah.
we're going to be tackling the mystery of red blood cell antigens
Okay.
and why they are so hard to study.
Right.
It's like a detective story,
Uh-huh.
but with tiny molecular structures instead of
I
fingerprints.
like that. Yeah.
And our source material for this deep dive
Okay.
is an excerpt from Chapter 3 of a scientific document.
All right.
And it starts by asking a really intriguing question, which is, what do we know about the Great Peptide Mystery?
That's a good question.
Yeah.
It really is a mystery
Yeah.
because, it highlights a problem that scientists run into all the time,
Yeah.
which is
Yeah.
you've got this antibody, and it's like our detective,
Right.
and it's trained to recognize a specific bad guy or antigen based on a mugshot. And the mugshot in this case is a synthetic peptide. It's like the simplified version of the real antigen. But sometimes the antibody sees that mugshot, thinks it knows the criminal,
Right.
but then completely fails to recognize the actual antigen
Oh.
when it's out in the real world, on the surface of a cell.
So it's like a case of mistaken identity, but at the molecular level.
Yes, exactly.
But why does that happen?
Well,
What's the difference between the synthetic peptides and the full proteins?
yeah. So think about it this way.
Okay.
The synthetic peptide
Yeah.
is just like a snapshot.
Uh,
It's a small piece of the bigger picture.
Yeah.
It's like a short chain of amino acids.
okay.
And it's designed to mimic just one specific part of a protein,
Got it.
but it doesn't capture all the complexity of the real thing.
Okay.
Proteins on cell surfaces.
Yeah.
They're like criminals trying to blend in a crowded street.
Right.
They're much larger, folded into all these intricate three-dimensional shapes,
Wow.
and they're constantly interacting with their environment.
Okay.
They can even have these modifications with other molecules, kind of like adding a hat or a disguise.
I see. So it's like our antibody detective is looking for a suspect with a specific facial feature.
Yes,
But then that suspect puts on a hat, all of a sudden they become unrecognizable.
exactly.
That makes sense.
Yeah, and you mentioned modifications to proteins.
Yeah, I did.
Can you think of an example?
Well, you mentioned like putting on a hat or disguise.
Right.
Is there something specific that they do?
Well, one very common one is called glycosylation,
Okay.
and that's basically the addition of sugar molecules to the protein.
So it's literally putting on a disguise.
It is, yeah.
Wow, that's wild.
It can change the protein's shape
Hmm.
and how it interacts with other molecules.
So even though the antibody recognizes that isolated feature,
Right.
it can't see it when it's part of this larger, more complex structure.
Yeah,
Wow.
that's the problem.
Yeah, and this must have implications for like developing antibodies for diagnostic tests.
Oh, huge implications.
For treatments, yeah.
Absolutely. I mean,
Yeah.
if we want to develop antibodies that work,
Yeah.
we need to understand how all these pieces
Right.
fit into the larger puzzle of the protein's structure.
So how do scientists even begin to study something so small and complex?
Well, it is really challenging,
Yeah.
but luckily they have some pretty amazing tools.
Mm.
In this case, our scientists decided to use something called in silico modeling.
In silico modeling. Okay, so
Yeah.
instead of working in a lab with test tubes and beakers,
Yes,
they're working on a computer.
exactly.
That's
So you can build like a virtual model of the molecule
right.
and then run all sorts of tests.
And see how it
Quote,
goes.
yeah, in different conditions and everything.
Okay, so what kind of molecule were they looking at?
So the scientist was interested in a protein
Okay.
found on red blood cells
Hmm.
called glycophorin A or GPA.
Glycophorin A, okay.
Yeah.
Why that particular protein? What makes that interesting to study?
Well, their goal was to understand a similar protein called glycophorin B, and that one has some important antigens on its surface. But GPA was a good place to start
Okay.
because it's related to glycophorin B,
Right.
and there was already some research on its structure.
Got it.
However,
Yeah.
even with that head start,
Yeah.
studying GPA is no walk in the park.
What makes it so challenging?
It's the disguise.
The disguise, right.
Yeah.
It's covered in sugar.
GPA is heavily glycosylated.
Okay.
All those sugar molecules can change its shape and
Right.
make it really hard to study.
Wow.
And then on top of that, it's also what we call intrinsically disordered,
Intrinsically disordered. Okay.
meaning it doesn't have like a fixed, rigid structure.
It's like a jellyfish or something.
It's kind of like trying to study a jellyfish.
Constantly wiggling and changing.
Exactly, yeah.
Oh, wow. Okay.
So you've got this slippery...
It's like a nightmare to study.
Yeah.
Okay, so I've got a slippery, shape-shifting protein covered in sugar.
Yep.
How in the world did they approach this?
Well, they ran a ton of simulations,
Right.
and they discovered something really interesting.
Mm,
It turns out that even though most of GPA is really flexible
right.
and disordered,
Yeah.
there's this one small region that forms a stable structure,
Okay.
and they call it a beta hairpin.
A beta hairpin. What is that?
Imagine taking a piece of string
Okay.
and folding it back on itself
Yeah.
to create this little tight loop.
Right.
That's essentially what a beta hairpin looks like in a protein.
Okay.
It's formed by two short segments of the protein chain that run in opposite directions,
Okay.
and then they're connected by a little loop.
So it's like this stable little paperclip
Yes.
in this like flexible string.
Exactly.
That's a great analogy.
And this beta hairpin is important
Mhm.
because it helps stabilize the protein's structure
Okay.
and may also play a role in how it interacts with other molecules.
With those antibodies.
Exactly.
Okay, so they found this stable region in this otherwise floppy protein.
Right.
What did they do with that information?
Well, that's where things get really interesting. But I think we should save that part of the story for part 2.
Ooh, a cliffhanger.
Yes.
All right, well, we'll be back for part 2 of our deep dive.
Looking forward to it.
All right, see you then.
See ya.
Welcome back, everyone. So in part 1, we were talking about this great peptide mystery.
Yes.
Where antibodies are trained to recognize specific antigens.
Right.
Using those simplified synthetic peptides.
Yeah.
But then sometimes they fail to recognize those same antigens out in the real world.
Yeah, it's like they can only recognize the mugshot, but not the actual criminal.
Exactly.
Yeah.
And we left off with our scientists discovering this stable region called a beta hairpin.
Right, within that floppy glycophorin protein.
Yeah, so what happened next? Did they try to use this beta hairpin
They did.
to create a better mugshot?
That's exactly what they thought.
Okay.
They figured if they could create a synthetic peptide that perfectly mimicked that stable beta hairpin structure, it might be a much better target for antibody development.
So it'd be like giving our detective
Yes.
a high-resolution 3D model
Exactly.
instead of that flat 2D mugshot.
Precisely, yeah.
Okay, so walk me through this.
Okay,
What
well,
did they do?
they started with like the simplest approach,
Hmm.
which was to just create a linear peptide.
Okay.
So it's basically just a straight chain of amino acids in the correct order to match that beta hairpin sequence.
So they had the right ingredients,
Yeah,
but not the recipe.
exactly. And not surprisingly, this linear peptide just kind of flopped around,
Oh.
failed to fold into the right shape.
So what did they try next?
Well, their next idea was to make a circular peptide.
A circular peptide.
Yeah.
Oh, okay.
So basically connecting the ends of the amino acid chain together.
To form a loop.
Yes, to form a loop.
Got it.
They were hoping that by forcing the ends together, it would kind of encourage the peptide to fold correctly.
Okay. Did that work?
Unfortunately, no. Even with this circular design,
Yeah.
the simulations show that the peptide still couldn't quite get it right.
Hmm.
It either like twisted into a different shape or kind of collapsed in on itself.
Oh, no. It's tough.
Okay, so 2 strikes. Did they give up?
Not yet.
Okay.
They had one more trick
Okay.
up their sleeve
All right.
for their final approach.
Yeah.
They basically decided to skip the whole folding process altogether.
Okay.
So instead of trying to coax the peptide into the right shape, they made a pre-folded beta hairpin peptide.
Whoa.
So this means they actually used some special techniques to synthesize the peptide in a way that it was already in the correct 3D shape.
So no assembly required.
Exactly.
Did it work?
It did.
Yay.
The pre-folded beta hairpin remained nice and stable, held its shape in the simulations. But
There's a but.
there's a but. Okay, this success came with a really important realization.
What was that?
They realized that this specific structure relies heavily on interactions with other parts of the full glycophorin A protein to actually maintain its stability. So it's not something that just easily forms on its own.
So even though they found this stable region,
Yeah.
it's not like you can just like snip it out
Exactly.
and it's going to act the same way.
Exactly. It needs that support.
It needs the context.
It needs that context, yes.
Okay.
And this has some big implications for future research.
Right.
Because it means that simply creating a synthetic peptide that matches the sequence might not be enough.
Okay.
You need to consider all those interactions
Right.
and potentially come up with new ways to mimic them.
Okay, so this research has really revealed a whole new layer of complexity
Oh, absolutely.
to this peptide mystery.
Yeah, but that's how science works.
Right.
Every discovery leads to more questions.
Yeah. New avenues to explore.
Well, this has been super fascinating.
I agree.
We've learned about all these challenges.
Yeah.
Of studying these tiny structures.
It's amazing what they're able to do, though.
Yeah, and all the creative approaches they're using.
Yeah.
To figure it out.
Totally.
Sounds like there's a lot more to learn.
Oh, for sure.
Well, we've reached the end of part 2 of our deep dive. So be sure to join us for part 3, where we'll wrap things up.
Looking forward to it.
And explore some of those potential avenues for future research.
Sounds good.
See you soon.
See you then.
Welcome back, everyone. So we've spent the last 2 parts of this deep dive really exploring this world of peptides and proteins and antibodies.
It's fascinating, isn't it?
It really is. And we've seen how scientists are tackling this great peptide mystery.
Yeah.
Figuring out why those antibodies sometimes struggle to recognize their targets.
Right, even when they've been trained on those simplified versions.
Like those synthetic peptides. And we left off with our scientists discovering that.
Yeah.
Even when they found that stable region within that protein.
It still needed the support of that environment around it.
Yeah, to actually work properly.
Exactly.
It's like one piece of a puzzle.
Yeah, it doesn't tell the whole picture.
Right. So where do we go from here?
Wow.
Knowing what we now know
I think
about this complexity, what are some of the paths forward?
one really exciting direction is to find new ways to mimic that natural environment of the proteins.
Okay.
But in the lab. So remember, they discovered that beta hairpin structure in glycophorin A, it needs that full protein to be stable.
Right.
So maybe we can find ways to create artificial environments that can do the same thing for these isolated peptides.
So it's like, instead of just giving our detective the one clue, we got to build a whole crime scene around it.
Exactly. Give them the full context.
Yeah, to help them solve the case.
It's like building a little stage set for our molecular actors.
I love that analogy.
So they can play their roles properly.
That's great. Are there any other approaches?
Well, another really promising one is to keep using those computational models.
Okay.
Because as computers get more and more powerful, we can create better simulations that take into account not just the structure of the peptide itself, but all those dynamic interactions with the things around it.
So we can really see it in action.
Exactly, like an interactive movie of the molecules.
Instead of just a snapshot.
Yeah, you got it.
Wow, so this research...
It's pretty amazing.
It could impact more than just red blood cell antigens.
Oh, yeah. This is much bigger.
It could affect all sorts of diseases.
Absolutely. This is about understanding the fundamentals of the immune system.
And that's a big deal.
Huge.
Wow. So it sounds like this is just the beginning
I think so.
of understanding this whole peptide mystery.
Yeah.
But it's really exciting to think about what could be discovered in the future.
You know, it's all about that scientific curiosity.
Yeah.
That drive to learn more.
Absolutely. Well, thank you so much for joining us.
Oh, it's my pleasure.
On this deep dive.
It was fun.
Into this microscopic world.
I love talking about this stuff.
Me too. And we hope you enjoyed it.
I hope so too.
And learned something new along the way.
Always learning.
So until next time.
Yes.
Keep exploring. Keep questioning.
Absolutely.
And keep diving deep.
I'll see you then.